Well, welcome everyone. We're glad that everyone's here in person or on Zoom. And this is sort of the inauguration of our seminars this semester. And it's I can't remember the last time we did one in person. So we're hoping to be able to manage the technology. A minute ago, we were trying to figure out how many PhDs it took to actually just get a flash drive to work. But we think we got that taken care of. Anyway, today it's my pleasure to introduce Jared Wilmoth. We okay?
Yeah, I just wanted to check on my phone myself. Please don't.
Okay. So, no, we're just making sure that the sound is coming through.
Yeah, the sound is coming through, it's just very.
Oh, so we're not getting it close?
We're not getting it from the board. So there's
We just want to make sure that it's, I mean, he's listening to it remotely and he's saying the audio quality is not very good. Okay. All right.
so
This is an experimental production event. But it is my pleasure to introduce Jared Wilmoth, who's our recently hired soil chemist. And Jared did his undergraduate work at West Virginia University and also did his master's degree there, during which time he was you'll be most happy to know that he was a member of the West Virginia soil judging team and traveled to, in 2005, traveled to California where West Virginia won the national championship. And for that year, they had possession of the trophy that we've had possession of for 3 years now, because of COVID, right? But we're gonna probably hand it back in the spring, or we're gonna try to defend it in the spring. Anyway, after that, Jared went on to the University of Georgia, where he did his doctoral work with Aaron Thompson. He finished up there and then did a postdoc at Princeton University. And we were very fortunate to nab him after that. So anyway, he has agreed to give us an update on some of the things he's working on, some of the things he's doing and thinking about. And so my pleasure to introduce, present to you, Jared Wilmoth.
So first off, thanks a lot for the introduction, Dr. Redox. That was great. And yeah, as he pointed out, that was like the first time in WVU history that we took the nationals. So that was a really great experience. And I was just circling around the trophy the other day, trying to find a year that we won the national. And so I'm looking around, I don't know if any of you, some of you were on the Lake with Marty during recent soil judging, and that's a great experience if you have a chance to take part in that. So for those of you who are tuning in from home, maybe a couple people could just send in a quick message to let us know if you can hear everything okay and see everything okay. This is our first seminar sort of kicking things off. So if you can get just a bit of feedback on how it's going in terms of the sound quality, the visual quality, that would be great. So this introductory slide for my talk today. So the title is Redox Heterogeneity, Entangles, Soil and Climate Interactions. And so I was basically hired and joined the department as a specialist in redox conditions in soils. Okay, so my field of expertise and what I'm really interested in are redox oscillations in different soil environments and how those drive different biogeochemical cycles that ultimately tie in with things like climate change. Okay, and so any system that has anything to do with redox oscillations, I'm really interested in. Now, over the summer, I was able to publish a paper. Actually, started writing it at the beginning of the summer and had it published at the end of the summer, which was, quite a feat. But it was a perspective piece that I wrote for a journal called Sustainability, and the icon for the journal is up in the upper left corner of the screen. But basically what I was trying to do in this paper was just think about the different interfaces, like the mineral, microbial, organic, rhizospheric interfaces over which redox heterogeneity operates in soils and how that can affect different things. Some of the key topics that I covered in that paper, if you're interested, were soil microsites, cryptic cycling, the Methane Paradox, which I'm going to talk about today, and soils. And one of the key concepts that I sort of end the paper on, it's sort of a concluding remark, and I just wanted to point it out here, was that soil redox heterogeneity at the level of minerals, microbial cells, organic matter, and the rhizosphere intricately regulates, yet also allows us to predict soil and climate interactions. So I'm going to talk a little bit about that today and why it's important. And so one of the things that I did for that paper was just try to think of a few things that climate change does, right? Like when the climate changes, what are some of the changes that we're interested in? Okay, changes in precipitation, frequency and magnitude, changes in wet-dry cycling in different soil systems, sea level rise, and all of these different types of things that the climate can do to hydrology in soil, for example, like in a wetland or in any type of soil for that matter. It can create oscillations, right? Or it can, or at least if the system is already undergoing some sort of cyclical sort of redox transition over time, climate change can perturb that oscillation, right? Either the frequency of it, the magnitude of it, and this can have huge impacts on things like methane emissions, which is what I'm going to talk about today. But of course, it could also have a big impact on things like microbial iron reduction or iron biogeochemistry, or just about, anything you can think of in the soil, redox has something to do with it, right? And so sort of the big idea that I was trying to figure out was, okay, or think about in this paper, was this idea of a climate feedback or a soil climate feedback, right? And this is something that happens when, so if we just start from soils, we know that soils emit things like CO2, nitrous oxide, and methane, and that these greenhouse gases fuel climate change, right? Now, you can imagine that this could just go in a feedback loop, right? So if climate pushes soils to produce more of these greenhouse gases, then these greenhouse gases will be emitted more from soils. They will feed back into climate, to different climate-driven perturbations in soils, and it could create just sort of like a fuel feedback loop. And so methane is part of that feedback. So the other thing that I tried to end that paper with are the different ways that we need to think about how we might characterize these complex processes and interactions, right? So I mean, some of the, and I tried to go for like these bigger sort of analytical methods that sometimes we don't tend to reach for as soil scientists or environmental scientists that are definitely out there for us to use. And some folks in the department are already using things like metaomics, metatranscriptomics or metagenomics, metaproteomics, metabolomics, those sorts of things to characterize microbiology. There's also some faculty that are using FTICR or cyclotron resonance, which is a really cool technique to be able to profile something like dissolved organic carbon. And you can simultaneously measure thousands of different molecules at once and get a lot of really in-depth chemical information. It's sort of like omics, except it's for carbon. And then there's synchrotron techniques that some faculty are also using actively to study minerals and carbon as well. But then for the, in terms of modeling, I know that there's a big interest right now generally for artificial intelligence, machine learning, deep learning, these types of things. But those are sort of, those aren't just singular types of modeling tools, right? Those are always sort of joined with, or they sort of enhance other types of modeling, tools like agent-based modeling, for example, which I've used in the past for some of my postdoc work, allows you to model the individual interactions between single microbes. And so molecular dynamics is another type of thing where if you want to model interactions between atoms or molecules, that's an even smaller scale type of modeling tool. So anyways, this is just food for thought. We can come back to this if anyone's interested in talking about it. But bottom line is, in order to study redox heterogeneity in soils, we really need to be grasping for these high power techniques, multifaceted, big collaborations, interdisciplinary. And so I was also fortunate. So Dr. Ray, of course, mentioned that they were able to nab me right after I completed my postdoc at Princeton University. And I was working at Princeton on this project of looking at methane emissions from wetlands. And so we were kind of exploring this counterintuitive process whereby oxygen can actually stimulate methane production. All right. So the redox heterogeneity part comes in where sometimes we think of wetlands as just sort of these on average anaerobic systems, but there is oxygen there, and sometimes it's intermittent, and that can lead to some really interesting biogeochemical cycling, which I'm going to talk about. And then methane, of course, if we can go back to that concept of a climate feedback, methane's part of that, right? So, this paper was published over the summer. So now I'm going to talk about the things that we did for that paper and what we discovered, and I'm going to put this into the context of where I'm taking a new direction here at the University of Maryland. So one thing that you need to realize is that methane emissions from wetlands make a very significant contribution. So if you look at a pie chart of all the methane emissions that go to the atmosphere, arguably wetlands can, if we consider the variability, and depending on what your definition of a wetland is and who you talk to, it can be upward of 1/3 of all methane emissions to the atmosphere, which again, methane is a more potent greenhouse gas than CO2, even though it's not as abundant, but wetlands contribute significantly to methane emissions. And so it's important for us to study wetlands from the perspective of a natural system, right? Because it's not anthropogenic, it's not human-derived methane, right? This is a huge slice of methane emissions to the atmosphere that's just being created by natural processes. So understanding natural processes in wetlands is really important. Understanding redox cycling in wetlands is really important for this. And so the classical view is that oxygenation tends to shut down methanogenesis, right? If you've got oxygen around, we typically don't think, again, traditionally, that you would see much methane being produced. And so this leads to, the other thing to realize is that methanogens can't handle complex carbon sources to produce methane. They really have to use very simple substrates like acetate, hydrogen, and CO2, and then C1 compounds like methanol to produce methane. And so in a wetland, it tends to store very complex forms of carbon. We might ask the question, well, what's one of the main drivers for methane production? If the carbon is really complex, and methanogens can't use complex carbon directly, there has to be some processing, right, to more simple forms. And so, again, the traditional view is that oxygen is bad for methanogens, and the traditional view is that methane is usually produced at least in saturated layers, strictly anoxic anaerobic layers, or in some cases, people might tend to think that it's produced in the deepest layers of the wetland. But then there's been a string of papers, not a lot, but some studies. So the first one that I'm indicating here was soil taken from the Waikia critical zone in Puerto Rico. So that's a forest soil in Puerto Rico, right? And then the second paper is from the wetland or wetlands in Vermont, I believe. And then the third paper from 2017, Nature Communications, was a study of a wetland around Lake Erie. And basically, whether you're in soils in Puerto Rico or you're in wetlands here in the northeast of the US, there's this observation that when oxygen's around, or if a soil undergoes redox oscillating conditions or transitions between oxygenated and not oxygenated, there's some connection there that tends to fuel methane production. So people have actually modeled and directly observed fairly recently that if you account for this type of process, that in several wetlands to date, that 60 to 80%, and that's kind of even 90% of methane coming from wetlands in some cases are actually coming from the near surface layers, where you would have a lot of oxygen around. And so it leads to a lot of questions, right? How is that possible? How would most of the methane being emitted from a wetland come from the surface where there's a lot of oxygen? Because oxygen is supposed to kill methanogens, right? And so really quickly, one of the things that might be going on, right, is you've got soil microsites that sort of shield the methanogens in an anaerobic pocket or something like that, even if the rest of the soil is oxygenated. But there could be something more mechanistic, right? Oxygen could somehow directly be impacting methane production. So, and that's kind of what we set out to explore. So in the traditional view, again, in a side-by-side comparison, the conventional model says that oxygen's bad, methane's only produced under saturated anoxic, strictly anaerobic conditions. But the emerging model is that if you have redox oscillating conditions, like where a water table moves up and down, right? This could happen in a tidal wetland because of tidal cycling. It can happen because of precipitation patterns or wet-dry cycling. But when you have that type of redox oscillation condition, it seems to suggest that you can get more methane produced by having oxygen in the system intermittently. And so our hypothesis was taking all of this into consideration because nobody had really nailed down a hypothesis who just stated this and like tested it in a mechanistic way. So that's what we set out to do. The hypothesis was pretty simple. It's that oxygen-anoxic or a redox oscillation or transition enhances methanogenesis. And again, we were trying to figure out, why do significant methane emissions originate from your surface layers where you have oxygen, and could oxygen play a more direct role in stimulating methane production? And so, what we hypothesized, like this is a visual hypothesis, is that so the big blue arrow is showing oxygen. Essentially, what we're showing is that if you have just an anoxic system, you will get methane production. But we believe that if you have oxygen in the system as well, that oxygen can promote the degradation of complex carbon. Remember, I said before, methanogens can only use relatively simple forms of carbon to produce methane. So to get to that simple carbon, you have to break down complex carbon, right? And oxygen is really great at doing that, right? Because it provides a really powerful electron acceptor for microorganisms to degrade or mineralize organic carbon. All right, and so oxygen could help sort of create this, you can't see it up here, but it says something about a carbon cascade. Well, actually, yeah, in yellow box. So you can almost imagine oxygen sets off a stronger chain reaction or carbon cascade leading to methanogenetic substrates. And so, in order to test our hypothesis, we used a pretty straightforward type of serum bottle incubation methodology. Went to the field, we collected wetland soils from a peatland, okay? And so it's a peatland that was derived under sphagnum moss. The pH tends to be pretty low, 4.0, even less than that, in some cases 3.7. And we collected the peat from different layers. And we wanted to bring it back to the lab and expose it to oxygen in a redox transition experiment, which I'll explain here in a second, to see how that impacts methane production. And so this is our experimental design. So keep in mind, we've got all these bottles filled with peat in the pore water that's found at the site. We've got different layers of peat, and we wanted to expose it to a couple different levels of oxygen. So we had this incubation experiment where all the soil was exposed to 232 days of just an incubation in the lab, okay? But a portion or our treatments were exposed to oxygen for roughly the first half of the incubation, and then we're allowed to naturally go anaerobic or anoxic again, just to see how that prior oxygen treatment impacted methane down the road, right, by the end of the incubation. And so we wanted to compare what happens when soil undergoes a redox transition with oxygen versus just a continuously anoxic system. So a continuously anoxic system is sort of like our control in this case. And what we found, very striking, is that even, we had different levels of degradation of the peat that we started out with, so we collected it from different depths. The things that look more fresh on the top, and then the things that are more black and graded were deeper from the profile. But what you can see is generally, regardless of the layer, so UNS would be the unsaturated layer, the layer closest to the top. at the water table layer is sort of like in the middle, and then below the water table would be the deeper layer. But what you can see is that soil treated with oxygen, like in that initial oxygenation step, when it was finally allowed to go anaerobic again and produce methane, it produced nearly 3 orders of magnitude, in some cases more methane than the anoxic controls, right? Which is shown on the right, the small columns. And so I guess I could back up and just say that I'm not going to show all of the chemical data that I have, but one of the strong things that we were able to pull out of this is that, and I'll show this in the microbial data in the next slide, is that the type of methanogen that we detected in the soil, it uses CO2 and hydrogen preferentially to make methane. So it's called a hydrogen-trophic methanogen, right? So you have to have CO2 and you have to have hydrogen to make methane in this type of system. And what we found is that when you add oxygen to the system, it tends to create like a pulse of CO2. And that CO2 that oxygen fuels, later that CO2 fuels methane production. That's part of the story here. So we used metagenomics and 16S gene characterizations, but long story short, the left column here just shows microbial groups, or it's actually phyla and a couple genera of bacteria that we detected that were really important. And as you go from top to bottom, so on the left are the types of microbes we detected. On the right are genes from those types of microbes that we detected in relative abundance. And so from top to bottom, we go from the microbes that you need to break down complex carbon, ultimately to Methanobacterium, which is the primary organism that we believe was making methane. Because all things considered, the way that it produces methane, based on other research as well, this seemed to be the smoking gun in our case. And so, to be more specific, by using metagenomics, we were able to tell that most likely the key genes, the key enzymes that are critical for the initial step of breaking down complex carbon in this peatland are these catechol dioxygenases, and then there's another one here that's phenol hydroxylase. You can't see the title, but phenol hydroxylase catechol dioxygenases. These are super important for the initial step of breaking down organic carbon. And then maybe I'll get back real quick just to point out that, like in the middle steps, this would be where that carbon that's been degraded in the first step, it has to be processed even further by different microbes. There's CO2 being produced all along this pathway, and then ultimately it's that CO2 that's fueling methane production and hydrogen. And so one of the ways that we wanted to characterize, to try to get a broad characterization profile of the organic carbon, the dissolved organic carbon fraction, we use this technique called Fourier transform ion cyclotron resonance mass spectrometry, or FTICR. And that's now other faculty, Dr. Yarwood's student actually presented some of this work, and I know Dr. Tor is also working with this type of data. It's very complicated, but it's also really great because you get a broad picture of the carbon that's in the system. So very quickly, I'll describe how the technique works. You basically take, in this case, it was just pore water samples. The pore water is essentially injected into a large chamber that is has a strong magnetic field. Your sample is ionized, and those ions are trapped in this magnetic field. And when they're exposed to radio waves, those ions travel in little packets in an orbit around detectors inside the instrument. And long story short, heavier molecules tend to travel slowly in the orbit, and lighter molecules tend to travel more quickly. And so the process, the software and everything, it can do a Fourier transform of that and basically figure out that, okay, the things that are traveling more slowly, those are your larger molecules. And so if you look at these mass spectra that I have in panels A and B, each one of those peaks, so on the left, toward the left, those are the small molecules, the things that are traveling quickly inside the magnetic field. And then as we move to the right, those are the larger molecules that are detected. Those are traveling more slowly in the magnetic field. Now, each one of those packets of molecules depending on how big it is, depending on how much of the different compounds are there, you get intense differences in the intensity of the peaks. And that can give you a sense of the relative abundance of the different molecules detected. Now, for me, the biggest differences that we saw with methane production occurred at the end of the experiment, okay? And so these are endpoints. Panel A represents a mass spectrum from a sample that's collected, and panel B also does, except panel A represents something that's our control, all right? So that was after a 232-day incubation under continuously anaerobic conditions. Panel B was after the oxygen treatment. And you can just see visually that the profiles are different, right? Now, one of the things you can do is for each molecule detected, for each peak, you can derive a chemical formula. The mass is so precise, you can derive how many carbons, how many nitrogens, oxygens, sulfurs, phosphoruses are in each molecule detected. And so you can make a diagram. This is called a Van Krevelen diagram or a Van Krevelen plot. And if we plot the ratio of hydrogen to carbon versus oxygen to carbon for each molecule detected, so forget about the colors now in panels A and B. The anoxic control molecules at the end of the experiment. So those are shown in blue. The molecules detected after oxygen treatment by the end of the experiment are shown in orange. And so when you overlay things like this, you can really get a good sense that, oxygen treatment, by the end of this incubation, even though both systems had undergone at least 100 days or so of anaerobic conditions at the end, prior oxygen treatment really caused major differences in the dissolved organic carbon chemistry. Now, I don't have time probably right now to keep going into that, but if you have questions, we can come back. So there's, because I know that there's other faculty members working with this type of data, I wanted to show a few different ways of looking at it. Okay, so you can plot the H to C and O to C ratios. You can also come up with a calculation called the nominal oxidation state of carbon, or the NOSC value. And that's really just a value that describes how oxidized the particular molecule is, the particular organic carbon. Now, the reason why that's important is because it tells you something about the thermodynamic properties of that molecule, how likely it is to be oxidized or reduced. And so there's some really cool calculations you can do for each one of the dots of each one of the molecules represented. And also you can plot those values against the exact mass, for example, of each one of the molecules. I should say that the red lines in each one of these figures just give you a visual reference point to sort of compare these data clouds between each other. And I should have pointed out that in each one of these slides showing these types of plots, that the left column is from the controls at the end of the experiments, the anoxic stuff. And then in the right column, this represents the treatment that was exposed to oxygen at the beginning. And so we can come back to this if anyone has questions, but DBE stands for double bond equivalence for carbon. So we can see that there was big changes in the double bond character of the carbon and the dissolved organic carbon fraction because of oxygen treatment. And then we can, the aromaticity index is just a measure of how aromatic the compounds are. So how polycyclic or ring structures that you would have in the molecules, there are big differences there. But the one thing that I really wanted to show today, and just kind of highlight quickly, was that you can also, remember I said you can get information on carbon, nitrogen, phosphorus, oxygen, hydrogen, sulfur. And so you can make plots with nitrogen versus carbon. Look at how that changes with the mass of the molecules. And again, you can see that for some reason, and I still don't have a great answer to this, prior oxygen exposure caused some unique partitioning between two distinct groups, apparently, of nitrogen containing organic compounds. And I have no idea why, but this might be interesting to take a look at or examine more closely in the future. So one of the other things that I'm interested in, okay, and this is sort of goes back to my PhD work. I know Dr. Reid, of course, is very interested in iron and manganese. And iron's great for studying redox condition or redox changes in soils because it's a very redox active species. So one of the things that we were kind of looking at but really just scratch the surface in the data set that I just described, was looking at how abiotic reactions with iron, so purely chemical reactions with iron, might actually be contributing to methane production. That sounds like kind of a far out idea right now, but it's actually pretty simple. So there's this classic chemical reaction called the Fenton reaction, where in the classical reaction, a catalyst like iron 2, so it's usually a divalent cation, but usually iron 2, reacts with hydrogen peroxide to create hydroxyl radicals. Now these radicals can attack organic carbon and essentially convert it chemically to CO2. And then, like I was talking about earlier, we have great evidence that microbes were creating CO2 that then fed methanogens. But in this case, we could also have a purely chemical pathway whereby iron catalyzes the production of CO2 through this Fenton reaction and degradation of organic carbon. And then that CO2 could potentially be used to produce methane by microorganisms, methanogens. So I just want to kind of talk just a little bit about this. Like I said, we just started to scratch the surface with it. This is something I'm still really interested in. We didn't have a conclusive answer. But going back to that one slide I showed, and I was talking about multifaceted analysis, one of the things that we used to try to map where the iron was in our peat samples. So we actually took some peat from our incubations. And this was actually a couple months into treating peat with oxygen. So this is under oxidizing conditions, but we were able to find these little, we were able to use the synchrotron radiation, synchrotron X-rays at a national lab to actually look for hotspots of iron concentrations in the peat. So all the blue area would essentially be organic carbon, okay, peat carbon. And then these hotspots would be the iron minerals that we were finding. We believe these to be nanoparticulate iron minerals, iron oxyhydroxides. That was confirmed with some Moss power data that I was able to do. But anyways, the main point is that during oxygenated conditions, there was iron 2 in the system. We were able to detect the reduced form of iron, which, you know, a lot of times I think we tend to think if a system is oxidizing, if there's a lot of oxygen around, that the chemical species will also be oxidized. That's not the case. Even when there's oxygen around or oxidizing conditions, you can have iron 2, and if you have iron 2 and you have oxygen, you can get these Fenton or Fenton-like reactions that could fuel carbon degradation and methane production. So it's not just microbes that are always involved with CO2 emissions and methane emissions from soils. Sometimes iron might be, could be just a purely chemical process that might be happening. And I'm going to end this little segment about iron. My slides got messed up a little bit in the wording, but that's okay. What this shows is that, one of the things I like to think about, and we've been talking about redox oscillating conditions, especially for iron, is that if you undergo these cycles of reduction and oxidation, depending on the frequency and magnitude of that, that can have big effects on whether we get more crystalline iron, whether we get poorly crystalline iron. And this is super important because there's a lot of studies, a lot of debate going on right now in soil science as to whether, or as to how important iron minerals are in their crystalline order in terms of protecting or shielding soil carbon, okay, from microbial degradation. And then also retaining nutrients, plant nutrients, agricultural fertilizer inputs, phosphorus, nitrogen. So understanding the impact of redox cycling on iron, whether it's a wetland, a peatland, or a farm's, farming soil, it's really important. And so I don't think I need to go through the conclusions of that research because I think I already went through that. The big conclusion was that oxygen causes the degradation of oxygen causes the degradation of complex carbon in a wetland. And then that complex carbon goes through a series of microbial steps leading to CO2 and hydrogen that then gets produced in methane. Okay? So oxygen is sort of a nice chemical catalyst to get this carbon degradation domino effect or cascade started, leading to methane emissions. Now, I'm going to show a handful of slides to finish my talk today. And this is sort of like a direction that I'm moving things into now. Still has to do with the methane work that I did previously, but it has to do with my passion for understanding redox dynamics in soils. But being here at Maryland now, I'm extremely interested in Chesapeake Bay wetlands and how those are contributing to climate change and how they're buffering climate change and how redox plays a role in that. And so, any of you that have been to the wetlands or taken a class in the department have probably heard of Phragmites. So Phragmites is this invasive common reed that has taken over a lot of wetlands, I believe globally, especially and also around Chesapeake Bay. This is something I'm still learning about. But the title here, Drivers of Redox Dynamics in Coastal Wetlands, these are very important drivers of redox dynamics. And this is something I'm really interested in, how the plants themselves could actually be driving these redox oscillations. So for my work in the past in the lab, I was essentially modeling soil redox oscillations by controlling oxygenation, no oxygenation in a serum level. Here, the plants are doing the same thing in the field. They're pumping oxygen into the soil and controlling the oxygenation and these anoxic-oxic transitions. But there's a lot of questions that I think no one's really looked at yet. So I'm really excited about this. I've been learning new things. Basically over the summer, I started some work with Dr. Baldwin and his graduate student, Sylvia Jacobson. And actually that's where this next picture comes from. It's one of the sites that they have at the Smithsonian Ecological Research Center. And then I started to get curious about different processes that might be happening related to soil redox. And there's some recent papers that I came across that really intrigued me. So number one, plants like Phragmites sort of create it's almost like plumbing, right? These reeds have essentially these hollow parenchyma tissues or their rhizomes. And we can almost imagine these as sort of pipes or conduits, right? And there's a really cool paper that came out in 2020 by Vandenberg et al. And they were sort of looking at how, okay, if you have methane produced in the soil, how does the sort of the pressure gradient that usually drives gas into the soil through Phragmites? So Phragmites use the pressure gradient to get oxygen down to the roots, and that's how the plant survives flooded conditions. The roots need oxygen, okay, to respire. And so the process of forcing gas or transporting gas to the roots and rhizomes actually creates a flow path to basically kind of any methane that migrates into the rhizome can actually get pushed out and then up to the atmosphere. Now, people have been actively studying gas transport to and from soils through plants, even trees, right? But this is a really cool paper that's sort of looking at a different thing where it's really the interconnections between multiple plants. And for example, in this case, you've got a broken stem at one end of the connection and a live plant at the other. And it could be, as it pumps gas into the soil, it's pumping other things out like methane and maybe nitrous oxide and CO2. But the thing that really captured my interest, right, the thing that really got me excited, and it's an area that I'm moving into now here at Maryland, is there's been a string of papers that have looked at how between night and day, diurnal cycles, okay? So during the day, the plants seem to transport most of their oxygen into the soil. And it kind of peaks by midday, right? Like the warmest part of the day generally. But then at night, it steeply declines, right? So you get oxygen pumped into the soil during the day, and then it basically goes away at night. And that could be through microbial degradation of the oxygen at night because the plant's not actively pumping any more into the soil. But in either case, you get these oscillations. And so the top lines that's being shown here, the dark line and the dotted lines that have this pretty sort of, kind of a sine wave pattern, right? So the dark line represents measurements that are taken sort of in the stem or the culm of a Phragmites plant. This was a different experiment, by the way, and it was in a constructed wetland, so kind of a synthetic wetland system, but nevertheless. And then the dotted line, the dotted sine wave, that's oxygen that was measured directly with a special type of sensor directly in the rhizomes. And so I just wanted to show that in the soil, in the rhizosphere, these plants are sort of modulating the redox conditions. And that could be really important, right? I've already talked about whenever you have these types of conditions, like I saw in the lab, you can enhance methane production by having these redox oscillations. So if the plants are in the field doing that same thing, this type of oscillation could be, there could be like huge spikes of methane occurring over relatively short periods of time, maybe that we're not capturing because of maybe it happens at night or maybe it happens under certain conditions, right? And while I'm on the topic, I just wanted to say too, that having a wave pattern or cyclical pattern happen like this in the environment is really interesting, especially in the wetlands, because you would also have tidal cycles that would be overlaying on top of these cycles. So you've got diurnal cycles of redox oscillation, you've got tidal cycles of redox oscillation. There's a lot of really cool things going on here. And just kind of, you know, it's my ideas going on. And so one of the ways that I want to investigate this, there's been a couple really cool papers. The ones I'm going to talk about here quickly is from 2018. But there's a special type of sensor called an optode, right? Now, optodes have been used for a while, and they make them such that they look sort of like a redox electrode or like a needle type of electrode, like a stick that you can stick into a plant tissue or the soil and get a measurement. But they have these camera-based optode sensors that allow you to get profiles. I think I'm just going to go ahead and skip this. So you can get these really cool. So this is a wetland plant that's growing in a wetland soil and basically a fish aquarium. And you can use this camera-based optode setup. So I'll explain how this works here in a minute, but there's these strips, or they call them foils. And so for example, this one is sensitive to oxygen, maybe. This one for CO2, and this one for pH, okay? And you can put those on the inside of the glass so that they're interacting with the system. And so that there's a transparent wall between the foil and the camera, all right? And this allows you to get maps, for example, of oxygen. That's what's being shown here. So we would actually have the ability, if you just go back real quick, we could have the ability to not just rely on kind of like an electrode type of measurement, right? Where you're sticking a kind of a needle sensor into a plant tissue and getting, you know, kind of like an average of a redox oscillation pattern. But you could actually map spatially how that's happening around the roots. And it's cool because these sensors can get pretty high spatial resolution data, spatiotemporal resolution. I think maybe it can get down to like 100 microns or something like that, which is really cool. And so, anyways, I think what I'll do is I'm going to finish up this data set and then I'll explain how optodes work really quickly. I think I have enough time. So here's the plant, here's the map of oxygen using this optode sensor. And then here's one of the ways, or here's some data that they got to show what happens between night and day around roots in this particular plant. Okay. And so I can't remember exactly how this was set up. It's either the same plant and they just chose like 5 different roots. Okay. I think that's how it was set up. Or maybe it was like 5 different plants and 1 root from each plant, something like that. But these represent different roots, these lines. Okay. So there's 5 of them here. 5 different roots were examined. They're showing one of them here at 6 different time points. And they're showing how during the night in the first step, okay, you don't see any oxygen around that particular root. During the day, it increases sharply, okay, and then during the next night cycle, during the subsequent night, oxygen drops off sharply, okay. So this is a really clear example of oxygen being modulated on a diurnal cycle. And this is really cool, right? Because who knows, because right now these papers have only examined sort of the mapping, right? It's just a proof of concept. Can we map this? What does it look like? But now the big questions are, what's happening with greenhouse gas emissions? How is this driving changes in the microbiome? Like, these are the big questions. And how is it changing the dissolved organic carbon fraction? And so these are big questions that I'm edging into now here at Maryland. I'm really excited about it. Now, really quickly, in just a couple of minutes, I want to explain how these octaves work, right? And then conclude my talk. So this, so I think in this particular setup, this is from a different paper, but it kind of shows very cleanly like how the setup would work. So you have, so first off, that red kind of rectangle that's inside of that brown square, that's sort of like where we've got one of these like aquarium setups or like what they call rhizobox. So you've got some transparent frame that's holding soil in a wetland plant. Inside of that transparent frame is one of those foils that I showed, right? One of those octave foils, one of these things. And the way that this works is you shine some particular frequency of light onto these foils from the outside. So here it's showing LED lights. Now, when you excite the foil with characteristic wavelengths of light, the foils will emit characteristic wavelengths of light that are filtered by the camera. Now that's real, so it's kind of like a fluorescence-based technique. I'm not exactly sure if it's technically fluorescence, but it's something like that. And so you're exciting these foils, and they emit light at a frequency that's picked up by, or it's filtered out by the camera. And the cool thing about it is each one of these foils is sensitive to particular analytes, whether it's oxygen, CO2, or maybe protons for pH, I guess. But essentially, you activate the chemical compound with light, and it emits light back to the camera that's filtered out. But the way that it actually detects the amount of the analyte, for oxygen, for example, would be the more oxygen that you have around the foil in a particular spatial location, it kind of quenches the signal, or it suppresses so when light gets emitted to the camera, if it was just under conditions without oxygen, for example, light would sort of slowly decay away from the foil as it makes it to the camera. But when there's an analyte around that interacts with the foil, that decay curve is even more steep. So the light coming to the camera decays way more quickly. Then the software program is able to convert that to a concentration of oxygen. Okay, so long story short, you can set up these experiments with a camera that has a special filter for the particular light that's excited and emitted to the camera. And then you can measure things like oxygen, pH, and CO2, at least right now. It'd be really cool if you can measure methane, but it's not quite there yet. And then you can get these really cool maps like this. And so imagine here we're looking at oxygen. You can also get maps of CO2. And you can also get maps of pH. Maybe one of these days we'll get maps of methane. But that's where I'm going to end my talk. So.
Time for a couple of questions for Jared.
Yeah.
Hi, so on slide 10, you talked about complex parts content being broken down by something.
Yeah.
Do you mean by broken down, are they broken down by aerobic microbes during periods where soil is aerobic?
Yes.
Okay.
Yeah, and that's a great question because you're exactly right. But and the question that I think is really interesting too is kind of along these lines. It's like, what do we actually? I mean, what does it mean for soil to be toxic or anoxic, right? And it's like, because I guess what I'm getting at is that let's say that the soil is mostly just anaerobic. Like let's say it's entirely anoxic for the most part on average. But you know, like I just finished talking about like the wetland plants that can like kind of pulse in oxygen between night and day. It's like, you know, it's a great question. Like, how do we want to characterize that? Like, maybe the best thing to say is just that it's an anoxic system on average, but then occasionally it gets oxygen, or it's a redox oscillating system. But you're exactly right. Whenever oxygen's around, the microbes, aerobic microbes, use that oxygen to break it out.
Thank you, Jerry. You put together a lot for us, and we really appreciate it. And we learned a bunch, including these kind of cutting edge emerging techniques. Super cool. You may not have shown it, or I may not have noticed. I was expecting to see some data that indicated that in the oxygen pretreatment, that you ended up with a suite of lighter carbon molecules. And was that data in here, or was it not?
Yeah, that's an excellent question. It's kind of why I didn't point it out specifically. And that's kind of why I'm glad that you brought us back to these. So I think it's a good way to. So whenever you look at these figures, like it's qualitative, right? But to answer your question, so I pointed out that you can make this calculation called the nominal oxidation state of carbon for each molecule is active. And that gives you an idea of how oxidized that molecule is. Now you can plot it against the exact mass, and the exact mass is, that kind of tells you how large or small the molecule is, right? So you're starting out with smaller molecules going to larger molecules, and then we're also looking at their oxidation state, basically, right? And so after prior oxygen exposure, if you look at this, I hope I'm right, I think I am, that they tend to be more oxidized, right? And they tend to be, if we look at sort of the centroid of the cluster of all these molecules here, it tends to be more, I don't know, around 450, whereas for the ones that have just been under anoxic conditions the entire time, the density tends to be shifted a little more towards 500. So exposure to oxygen leads to a large population of DOC that is slightly smaller and more oxidized compared to an anaerobic control. So this is one way that you can see that sort of information.
And so another thing that I've learned from this talk is that the oxygen that is brought down into the rhizosphere by plants, that it's not through this passive connection that exists 24 hours a day. It's just not oxygen from the atmosphere diffusing. It would rather seem to be oxygen that's produced in photosynthesis is what's brought down to the rhizosphere. Is that correct?
This is a great question because it's something that I kind of like was trying to figure out throughout the summer, and it really depends on the plant. So Sylvia is an audience, and she might be able to speak to this. But so I know for Phragmites and Typha or cattails, they both have more of an active transport of gas, where they have a pressure gradient that more sort of actively gets gas to the roots and rhizomes, and some of that ends up in the rhizosphere, like in the soil. And so yeah, that's a more active way to get gas, like in those plants, right? But then there are other plants where, sort of like you were saying, it's more passive diffusion. And yeah, I mean, so like I said, yeah. I feel like I lost my train of thought. I don't know that question. Like, so could you ask me that question again, David, please? Because I had a great answer for it and I lost my train of thought.
Well, it's great because it was one of the things that I could have, I think I might have learned from this. Is that not all, number one, not all plants are the same. I thought that the case for all wetland plants with arrangement tissue is that it's just a very passive process, whether it's day or night, oxygen levels in the atmosphere are basically the same.
Yeah.
And so therefore I would have expected, similar oxidation of the rhizosphere, no matter what the plant was. But you're saying that some plants are actually literally pumping it down,
Yes,
which
yes.
was a surprise.
Yeah, and that's
that you got a diurnal oscillation.
Yeah, no, I just caught my train of thought really quick when I answered this question, because you asked about photosynthesis the first time you asked. And that's what I wanted to answer. So Phragmites and maybe even cattails, it's really driven by this pressure gradient. And the oxygen is not, based on what I've seen, most of the oxygen appears to be coming from the surrounding atmosphere, not from the plant internal tissues during photosynthesis. Whereas for another plant, like Lobelia dormata, so this is an aquatic plant, right? It tends to be more flooded, like in the area it grows. But something like this may be more passive, where the oxygen diffuses down during the day. Most of the oxygen may be being produced from photosynthesis in the tissues, diffuses down into the roots and rhizomes, into the rhizosphere, and then during the night, that kind of turns off. And then the microbes in the soil at night kind of deplete the oxygen that's there, create CO2. And then it's actually kind of cool because what these papers are showing is that the CO2 that gets generated from the oxygen from the plant, that That oxygen gets converted to CO2, and then that CO2 can then be used by the plant the following day during photosynthesis. So it's a really cool process. And it depends on the spatial distribution as well, how far away you are.
So because plants matter, the degree of invasion by exotic plants could conceivably change the function of a weapon during the creation of this methane.
Yeah, exactly.
You know,
Which leads to weapon management. And one of the ways that we might be able to control greenhouse gases in wetlands, or like one of the things we should definitely be thinking about as we construct wetlands or manage them, is what types of plant species could sort of help mitigate carbon emissions.
Well, great questions. I'm sure there's more questions out there. I think we should formally draw this to a close. And I'm sure Dr. Wilmoth will be glad to chat with anyone further after this. So let's thank him one more time.